In this video, we will discuss the questions related to the recombinant DNA technology. We look into the questions from the workbook biology, question number 8. The questions is related to the restriction site and the restriction enzyme that will recognize and cut the specific sequence of the DNA. Figure 8a shows strand P and strand Q. Now we will look into the questions. Question 8a, it's about the restriction site and the restriction enzyme. Now we will answer the question. Question number 8a1. Identify the restriction enzyme which cut P and Q. For P, we have the palindromic sequence 5' GAE-TTC. This restriction site will be recognized and cut by restriction enzyme E-CoR1. While for strand Q, the palindromic sequence consists of 5' CCC GGG 3'. This palindromic sequence will be recognized and cut by SMA1. For the next questions, how many pieces of DNA fragments produced by cutting P with restriction enzyme? So we will have 3 fragments when we cut the strand P with E-CoR1. For the following question, list the sticky ends base sequence of clip P and Q. For strand P, the sticky ends consist of 5' AATT 3'. But for Q, there will be no sticky end because strand Q will be cut by SMA1 that will produce blunt ends. The next questions, what would happen if restriction enzyme failed to function? The natural function of the restriction enzyme within the bacterial cell is to cut the viral DNA. If the restriction enzyme becomes non-functional, it will unable to cut or to degrade the viral DNA. So the bacteria become unable to defend against the viral infection. Question 8b related to the screening process that involved the use of antibiotic and X gel in culture medium. The table 1 shows the number of bacterial colonies in 3 different types of culture medium. 50 colonies in the original medium, 10 white and 30 blue colonies in culture medium contain ampicillin, and there will be no survived colonies in the culture medium contain tetracycline. Let's answer the question. Question 1. Identify the screening technique. The screening technique is blue-white screening. The next question, why the tetracycline antibiotics inhibits the growth of bacterial colony? Bacteria usually contain plasmid with ampicillin-resistant gene, but has no tetracycline-resistant gene. The bacteria will not survive in culture medium contain tetracycline antibiotic. The following questions, what is indicated by white colonies? The white colonies consist of bacteria with recombinant plasmid, that is, plasmid that is inserted with foreign or target gene. Explain white colonies. White colonies consist of bacteria with recombinant plasmid, target gene inserted into the multiple cloning site of the plasmid that will interrupt or replace the LAC-G gene to become non-functional. So the LAC-G gene not able to code for the enzyme beta-galactosidase, and the bacteria will unable to hydrolyze the X gel. This will form white colonies bacteria. Question number 8c related to the step involved in synthesis of human insulin using cDNA and gene cloning method. The figure 8p shows the step in synthesis of cDNA and isolation of plasmid, followed by the sequences of processes to form recombinant DNA. Question number 8c1. Identify molecule X and Y. Molecule X will produce cDNA. So molecule X is mRNA. Molecule Y is a type of cloning vector. So Y is plasmid. The consequences when the enzyme reverse transcriptase or DNA polymerase become non-functional. Both enzymes unable to carry out its normal function. The non-functional reverse transcriptase unable to carry out the reverse transcription process. There will be no first strand of DNA will be formed. The non-functional DNA polymerase will not able to synthesize the second strand of cDNA. In short, cDNA will not synthesize. How sticky ends are linked between cDNA and plasmid. There will be two bonds will form. First, the strong covalent bond that will be called phosphodiester bond will be formed between the sugar phosphate backbone between cDNA and plasmid. The second bond that we call hydrogen bond will form between nucleotides of complementary strands to form the recombinant plasmid. The last question, the benefit of insulin produced using cDNA. Insulin produced using cDNA compatible to human that has no adverse reaction. It also can be produced in large amount, cost efficient, and it is halal. You can refer to the following suggested answer for your reference.